Living in space is hard on the human body. We evolved in our comfortable earthly gravity of 1g, but in microgravity, where we are virtually weightless, our physiology undergoes major changes in response to the lack of gravity. We haven't evolved any kind of anatomical features to deal with life in microgravity, so our bodies respond in a markedly physical, mechanistic way. Without gravity compressing us, our vertebral discs can expand a little bit, and so in space, the human body gets taller. Fluid also behaves differently in microgravity. The eyeballs change shape. They become more spherical in response to the fluid inside, and this distortion of the retina creates blurred vision and difficulty focusing on particular objects. Our blood normally pools near our legs when we stand upright on Earth, but in microgravity, the blood will instead diffuse more or less equally throughout the body. The legs will hold less blood than normal, making the backflow valves in the veins almost redundant. At the other end of the body, the head will hold a lot more blood than normal. In space, the head is almost swollen with fluid, with the veins at the temples enlarged and quite visible, almost like the person is upside down back on Earth. From a biochemical perspective, life in space can be quite dangerous. Our immune systems experience hiccups, and exposure to radiation puts our cells at much greater risk of mutation. With no gravity, the bones are under much less stress, and they begin to dissolve and rapidly lose their density. Muscle tissue is also under much less stress, and muscles will atrophy even faster. Now here's the big question that's the focus of a lot of recent research, and the big topic that I wanted to bring to you today. How does reproduction work in space? Not just the physical act of sex itself, but the chemistry of fertilization and gestation in 0G. This particular aspect of human physiology in space is largely an unknown frontier. Now, NASA has sent at least one married couple up into space, and all of the single people that are together in tight quarters are known to get very familiar with each other. So it's distinctly possible that humans have at least practiced the first step of reproduction in space. But to date, no one has gone through a pregnancy in space. And this is where we hit a bunch of unknowns. We don't know how pregnancy works in space, or if a woman in microgravity can even get pregnant in the first place. Early experiments with animals like roundworms and frogs showed promise. These animals were able to reproduce in space, and the offspring were healthy and normal. Development in 0G didn't really seem to have harmed them. But later studies on this same topic were a lot less successful, especially studies with rats, many of which resulted in rat populations that had not fertilized any eggs at all. This is very strange, as rats are usually very fertile and very eager to have sex, and they reproduce rapidly. But in these small population groups that were sent on space missions, they didn't have sex, they didn't reproduce. Joseph Tash is a reproductive biologist at the University of Kansas Medical Center, and he's done research on these space-bound mice, and is currently doing research on human gametes in space. When studying these various rodents, he noticed that the female rats who went to space in 2010 and 2011 all had diminished expression of the corpora lutea. The corpora lutea are short-lived glands that emerge in the ovaries, where they produce sex hormones and provide nourishment to developing embryos. But in these female rats that went to space, they didn't see any corpora lutea activity at all. Mr. Tash said, quote, "We found that in all the female mice that were exposed to spaceflight from 12 to 15 days, the corpora lutea were either totally absent or there were very, very few," unquote. This is not good, because the failure of these corpora lutea structures to emerge and play their role in early gestation is a deeply rooted biochemical reproductive barrier that's present very early on in the process. More recently, Mr. Tash has been doing research on human sperm cells that have been stored on the ISS, the International Space Station, for certain lengths of time. The idea here is to see if sperm cells can function properly in microgravity. Will the sperm structure, including its head and tail, be formed properly without gravity? Can they effectively swim in 0 G? The answers to these questions are buried in the mountains of data that have been generated during this research, and Mr. Tash and his colleagues are sifting through that data right now. They also have future experiments that are planned, including a 30 to 37 day trip in space for female mice, scheduled for next year. This field of study is extremely fascinating, and it's also extremely important to the future of space travel and space colonization. If we can successfully reproduce in space, then space colonization has become a lot more realistic. But if we can't, if some quality of microgravity makes it impossible to carry a healthy pregnancy to term, then human colonization of space becomes much more difficult. This is the kind of research that teaches us which doors are open and which are closed, ultimately informing us on how to spread out beyond the Earth and escape extinction by spreading into the cosmos.